afternoon, everyone. And I hope everyone spent a Merry Christmas break yesterday. My name is Jestlin, and I'm a current master's student in Ajuda학교 University. Firstly, I would like to thank the BRIC team for the opportunity to present in today's webinar. Today, I'll be presenting about our research titled HepScope, CNN-based single-cell discrimination of malignant hepatocytes. This paper was actually published in October this year in the Journal of Big Data. So in today's presentation, we will first explore the limitations associated with traditional biomarkers or markers of HCC. It will then be followed up with the development and validation of our novel gene set called HepScope and its ability to diagnose malignant hepatocytes and HCC. We will then explore how HepScope is used to develop an AI model to help predict for malignant hepatocytes from non-malignant hepatocytes. And finally, we'll then explore how HepScope has an influence on the tumor microenvironment. So firstly, let me briefly go through why we decided to research on HCC. Liver cancer is actually one of the top leading cause of cancer deaths worldwide, and its incidence is actually rising constantly, probably due to aging and population growth. And in Korea, about 15,000 cases of liver cancers occur in a year. And the common risk factors associated to liver cancer is hepatitis viral infection, fatty liver disease, or excessive alcohol consumption. When it comes to liver cancer, there are various subtypes arising from different cell types. Hepatocytes actually makes up bulk of the liver, and cancers that arise from these cells are known as hepatocellular carcinoma or HCC. And cancers that arise from the bile duct cells are known as cholangiocarcinoma, while rare cancers that arise from the veins or the arteries of the liver are known as angiosarcoma or hemangiosarcoma. So since HCC actually accounts for more than 80% of liver cancer, our research decided to focus on this subtype of liver cancer. So the main treatment options for HCC include definitive therapies such as whole or partial hepatectomy and liver transplant. When surgical treatment is not an option, radiofrequency, microwave ablation, or transarterial chemoembolization are performed. Liver transplant and ablation are actually potentially curative with a median survival of about more than 30 months, but typically it is only applicable for early stage patients because of the tumor size and the number of nodules. However, in liver cancer, actually most patients are diagnosed at the advanced stage due to the lack of tumor-specific symptoms. For this patient, systematic therapy are mostly applied. Up until recently, the first line of systematic therapy would include sorafenib and maybe some other multi-kinase inhibitors. However, we can see that with sorafenib, the median overall survival is actually less than 1 year or just slightly above 1 year. And we can see that these are numbers in Korea. Even in the past decade, the number of liver cancer deaths remains at 10,000 or more and does not decrease below that. So recently, there has been the introduction of immunotherapy with the use of atezolizumab or bevacizumab. And even with this immunotherapy introduction, the median overall survival is less than 2 years, so about 9 months extension from sorafenib. And we see that from all these efforts, the therapeutic outcomes for HCC patients still remains quite unsatisfactory in the field, and resistance to treatment is actually quite frequent. So one way to improve treatment is the use of biomarkers, either for the early detection of HCC or to predict for treatment outcomes. But in HCC, it is actually one of the cancers with a lack of such biomarkers. And the only widely used biomarker clinically is AFP. And even so, AFP is known to be elevated in non-HCC conditions, such as patients with hepatitis or liver cirrhosis. So it lowers the specificity. And also patients, not all patients with HCC actually has increased AFP level. We can see here from the very low, quite low sensitivity. So in diagnosis, people will usually use AFP together with ultrasound or some imaging tool to diagnose for liver cancer. So with the lack of biomarkers, there is actually no nation screening-wide program established in Korea, and it's actually not established in most countries. And it explains for why most liver cancer cases are diagnosed in the advanced stages. So our research actually focused on hoping to be able to contribute to this HCC biomarker research. So when it comes to biomarker discovery, bulk transcriptomic signatures have typically been used to determine for prognostic biomarkers in cancer. However, bulk sequencing lacks the resolution to capture cell populations of tumors and their complex microenvironment. Fortunately, with the development of single-cell RNA-seq, it allows for the classification of cells on the single-cell level, which helps to define more precise prognostic and diagnostic biomarkers in cancer. So in our paper, we exploited a large single-cell database that was curated for liver cancer called GetLiver. In this database, there was 17 data sets that were integrated, which amounted to a total of 410,000 cells. And from this database, as we are interested in hepatocellular carcinoma, which arise from the malignant transformation of hepatocytes, we extracted hepatocytes from normal, adjacent HCC, and HCC tissues. So to identify for biomarkers in HCC, we want genes that are expressed in malignant hepatocytes, but not in the non-malignant hepatocytes. So the classification of these two types of hepatocytes are very important for downstream analysis. From the extracted data, there were a total of 34,000 hepatocytes, which were classified into malignant and non-malignant hepatocytes using a tool called InferCMV. So this tool actually predicts for copy number variance, as we know that malignant cells or tumor cells will have more aberrant copy number variance than normal cells. And from this UMAP plot itself, we see that the annotated malignant and non-malignant hepatocytes are not separated well. They're quite in a similar region. So simpler clustering might not have been able to separate these cells from each other well. However, most papers actually don't run this inverse CMV tool to classify for malignant and non-malignant hepatocytes because it's actually quite computationally and time-consuming. And when running for inverse CMV, normal cells samples are actually needed to be used as a reference. So this is not a resource that most researchers would have, I would say. So many papers actually just use the expression of common HCC markers out there to annotate for malignant hepatocytes. So here, we have actually curated a list of the 11 common HCC markers used by other papers when they annotate for malignant hepatocytes on the single cell level. So when we verify the expression of these 11 common markers between the malignant and non-malignant hepatocytes annotated by InferCMV, we see that the expression of the 11 markers are not so different between the two groups of hepatocytes. And we also collected spatial transcriptomics samples from HCC patients. Here we also see that the 11 markers are not so consistent between the samples. And we see here that the AFP, the clinically used AFP, is actually not expressed in all tissues on the malignant hepatocytes and is in very low numbers, even in the single cell RNA-seq data. So next, we search for publicly available HCC gene sets on the MSeq database, which were actually described to have elevated expression in HCC tissues as compared to normal or adjacent HCC tissues. And we found 5 of such gene sets. Note that these gene sets were actually derived from analyzing the expression on bulk transcriptomics level. So when we validated this in our single cell data set, we calculate the module score or enrichment of these gene sets in the single cell data, and we see that these publicly available gene sets are not able to separate between the malignant hepatocytes from the non-malignant hepatocytes. Yeah. So hence, we actually developed a new gene set called HepScope that is able to separate between the malignant and non-malignant hepatocytes. To generate for this gene set, we leveraged the 34,000 hepatocytes from the GAP liver database and performed differential expression analysis. Genes that met the criteria of log2 fold change more than 0.6 or p-value less than 0.05 were selected to form the HepScope gene set. And this gave us 113 genes that were significantly upregulated in the malignant hepatocytes as compared to the non-malignant hepatocytes. And when we compared the HepScope gene set to the publicly available HCC gene sets out there, we see that more than 60% of the genes are novel, which is about 77 genes out of the 113 genes. Sorry, my images are really big, so it's kind of lagging. Okay. So here we show that the module score of our HepScope gene set are more highly expressed in the malignant hepatocyte than non-malignant hepatocytes in the GAP liver data set, which is what we expect because we got it. That is our reference data set. But we also validated it in a separate data set from SC-Atlas. And we see that in the module score is higher in the malignant hepatocytes than in the non-malignant hepatocytes. And once again, in this SC-Atlas, the hepatocytes were actually separated by inference CMV as well. So this is a heat map also showing that, once again, the other gene sets are not able to separate malignant hepatocytes from non-malignant hepatocytes, but our HepScope gene set module score is able to do so. And next, we also tried to check its translatability to spatial transcriptomics data. So firstly, in COSMICS data, which allows for single cell resolution in spatial transcriptomics, there is only a limited number of genes that can be sequenced with such techniques currently. So in this publicly available data set, we were able to find 22 genes out of our 113 genes, which is a limited pool. But even with the limited pool of genes, our HepScope gene set was able to separate HCC tissues or malignant hepatocytes from the normal tissues itself. The radar shade is a higher module score of the 32 genes. Then next, we looked at the vision data, which we lose single cell resolution, but allows for sequencing of the whole transcriptome. So here we show the module score of the 113 genes, all 113 genes. And we see that the module score was higher in HCC tissue than in its adjacent HCC tissue within patients across different patients as well. Then we move on to validation in the bulk transcriptomics level. Analysis was actually performed in 1,309 tissues from 21 data sets, including tissues from 362 normal samples, 256 adjacent HCC samples, and 691 HCC samples. And HepScope genes individually could effectively distinguish between HCC and normal or adjacent tissues. And GSCA analysis also shows stronger enrichment in HCC samples when compared to adjacent HCC or normal samples. And when we look at the single sample enrichment or SSGSCA, HepScope demonstrated once again that it can diagnose for HCC tissues as compared to normal or adjacent tissues. And it actually shows better performance in distinguishing tissues when compared to the Midorikawa gene set. And also there's a reduction in batch biasness when it comes to using HepScope when compared to the ex vivo gene set. So next, to assess HepScope's prognostic potential, we actually generated a risk score formula using the Cox regression coefficient of each HepScope gene using the TCGA data, and is validated in this separate data set here. So this score was actually able to stratify patients into low and high risk group, whereby high risk patients have a worse overall survival in both the TCGA and the validation data set, as well as a worse disease-free survival. And we perform univariate followed by multivariate Cox regression analysis, and we see that the HepScope risk score was the only independent predictor of survival in our risk scoring system. So up until now, I've shown you how HepScope module score or its enrichment is able to diagnose for HCC or malignant hepatocytes. But such methods looks at the average expression of our gene set only. So we wanted to leverage the distinctive expression patterns of each 113 genes within the gene set for even more precise prediction of malignant hepatocytes. So to do so, we actually thought of using the AI tools to help with complexity of working with multiple genes. And many of you would know that the use of CNN models are used to develop AI tools related to speech recognition or electrocardiogram data. But when we look at the input data, we actually happen to observe structural similarity between ECG data with our gene expression matrix. So we opted to employ this 1D CNN model commonly used for such time series data for our analysis. But the key aspect here in such models is that there is a fixed time point for each of the input data. In our expression gene set, there is no such time point. So to mimic this structure, we actually ordered the HepScope genes based on their descending log2 fold change, so that our input is the same for each cell. We then built our HepScope model, which comprises of three 1D convolutional layers. The layers uses ReLU activation function, and there is max pooling for downsampling. There is batch normalization for stabilization, and there are dropout layers to avoid overfitting. The output is then fully flattened and input into two sets of fully connected dense layers with the CeLU as the activation function, and also and a sigmoid activation function as well. So CeLU is actually selected to prevent the vanishing gradient problem and to make learning more efficient. Comprehensively, we then trained and tested the model by using the normalized gene expression matrix from the 11 single-cell RNA-seq datasets. So to ensure the sample number balance from each class, we perform random sampling on the malignant hepatocytes. And in total, the model was trained with 8,000 cells and validated and tested with 4,000 cells. So we then demonstrate that the HepScope trained model outperformed the same model but trained with different HCC gene sets. We showed that HepScope performed better using the AUROC and AUPRC scores, which is one of the highest is the highest amongst the other gene sets. Next, we then benchmark our HepScope model against other AI-based models. So we use this model, we got these models and trained them using our HepScope genes to show that our 1D CNN model was more superior. So for models that were 2D convolution layers, we converted to 1D convolution layers and standardized the hyperparameters to ensure fair evaluation. And HepScope actually demonstrates superior performance and balance across the evaluation metrics. I know that here the VGG16 has a very high recall, but when we look at the other matrix as well, it's difficult to say that the VGG16 is more convenient than the HepScope model. So in the last part of our analysis, we wanted to see if HepScope could have an influence on the tumor immune microenvironment. As we know, the interaction between cancer cells and their microenvironment influences cancer progression. Such interaction can actually result in the inhibition of anti-cancer response by activating and mobilizing immune cells with immunosuppressive properties. So tumor cells can adapt the immune evasion mechanisms and promote the reprogramming of the immune cells to support tumor development. And the infiltration of immune cells leads to the imbalance of the chemokine and cytokine production to generate reactive oxygen species, reactive nitrogen species to promote the development of fibrosis, cirrhosis, and in long term, cause the transformation of cells into a malignant state. So in HCC particularly, this formation of such tumor microenvironment is actually critical, and it affects the emergence, progression, and response to therapies, especially with the rise of immunotherapy as a treatment option in HCC. So the tumor immune microenvironment in HCC is very sophisticated. It consists of very diverse cellular and non-cellular components. The cellular components can comprise of various immune cells like the macrophage, neutrophils, myeloid-derived suppressor cells, your natural killer cells, which are what we want in the cancers. And it can contain like non-immune cells like cancer fibroblasts, cancer-associated fibroblasts, and also, and also like the extracellular matrix, which is non-cellular and soluble factors that are secreted by the tumor cells and stromal cells. So to investigate for how different expression of the HepScope genes may influence the tumor immune microenvironment in HCC, we actually stratified the 121 HCC samples based on ascending and HepScope module scores, and samples that were in the top 25% were classified as HepScope high samples, and samples that were in the bottom 25% were classified as HepScope low samples. We then calculate the proportion of the various immune cell populations in these samples. And we can see here is quite interesting that we see that HepScope high samples have an increase of CD4 T cells. And even for the plasma cells, there was an increase in their proportion. The macrophages remain quite similar, but we will now see that in the next slide, when we further classify the immune cell subtypes, in the CD4 T cells that were almost increased 2 times in the HepScope high samples, the Treg cells were actually 2 times higher in the HepScope high samples. And for the macrophages, although in the level 1 annotation, the macrophage percentage did not increase so much in the HepScope high samples, the tumor-associated macrophages had an increased proportion, which indicates that out of the macrophages in the HepScope high samples, there were more tumor-associated macrophages. And in contrast, there was a decrease in cytotoxic T cells in the HepScope high samples. So this shows that the HepScope samples that were classified as HepScope high had a more immunosuppressive tumor immune microenvironment. And finally, this is my last slide for my summation. We actually performed cell-to-cell interaction analysis using NicheNet to observe for interactions between the immune cells to the malignant hepatocytes between the two groups, HepScope high and HepScope low. So NicheNet analysis actually predicts for signals that may biologically influence the cells by checking the expression of genes in the pathways that are downstream of the signals here. So in this result here, we observed that in the HepScope high niche, the signals that were predicted to influence the malignant hepatocytes biological behavior are predominantly associated with ECM or the tumor promoting signals. And at the time of our research itself, we noticed out of these signals here, the lymphotoxin beta signaling was from the Treg cells to the malignant hepatocytes in the HepScope high niche, but it did not occur here in the HepScope low niche. It was actually quite novel at that point of time in HCC. It might have been investigated in other cancers, but in HCC, there has not been such research at that time. And over here, even in the HepScope high niche, we see that this signal was quite loud, which means that the LTB and LTBR genes were quite highly expressed in the ligand expressing cell, the Treg cells, and the receptor cell, the malignant hepatocytes. So this signal is known to be related to the NF-kappa B signaling pathway, and is actually shown to induce the migration of Treg cells, regulatory T cells. So it definitely encourages for more research. But all in all, this tumor immune microenvironment analysis suggests that HepScope may not only predict for malignancy, but also can shed light on the tumor immune microenvironment in HCC. So in summation, the HepScope gene set, which features 113 genes, represents a significant advancement in understanding the complexities of HCC. Its utility extends beyond identification of malignant hepatocytes on the single cell level, as we see its translatability into spatial and bulk transcriptomics data as well. And it also encompasses prognostication and capability, and is also able to categorize the tumor immune microenvironment between patients. So moreover, our study actually pioneered the use of the convolutional neural network model for gene expression analysis in HCC. Although our study have some groundbreaking findings, there remains room for further validation by conducting experimental validation of the LTB or LTBR signal, which will definitely strengthen the impact of our discoveries. Yeah. So I extend my gratitude to my colleague, Yoongjin Go, who is my shared first authorship, and also to Professor Darren Wontek Lim and Professor Subin Lim for their invaluable guidance and support throughout this endeavor. And I'm also grateful to my lab mates here for their constructive and continuous support, and I welcome any inquiries or opportunities for collaboration via email. And yeah, that brings me to the end of my presentation. Thank you for listening, and I'll take any questions from the floor.